Hi everyone. So, in our topic of spatial senses, we do have a question about visual pathway. But before going to that, we should have to learn about the visual process. So, we are taking this today, and maybe this question can be asked in exams in 5 marks, of course. So, again, it is important. So let's start this. Visual process is a series of actions that take place during the visual perception. During visual process, image of an object seen by the eyes is focused on retina, resulting in production of the visual perception of the object. When the image of an object in environment is focused on retina, the energy in visual spectrum is converted into electrical potentials, or we can say it impulses, by rods and cones of the retina through some chemical reactions. Impulses from rods and cones reach the cerebral cortex through optic nerve, and the sensation of vision is produced in cerebral cortex. Thus, process of visual sensation is explained on the basis of image formation and neural, chemical and electrical phenomena. Image forming mechanism. While looking at an object, light rays from the object are refracted and brought to a focus upon retina. Image of the object falls on the retina in an inverted position and reversed side to side. In spite of this, the object is seen in an upright position. It is because of the role played by the cerebral cortex. Light rays are refracted by the lens and cornea. Refractory power is measured in diopter, which is also symbolized as D. A diopter is the reciprocal of focal length expressed in meters. Focal no matlab apne lai shake relating to the focus of lens. Focal length of cornea is 24 mm and refractory power is 42 D. Focal length of lens is 44 mm and refractory power is 23 D. Neural basis of visual process. Retina contains the visual receptors, which are also called light sensitive receptors, photoreceptors, or electromagnetic receptors. Visual receptors are rods and cones. There are about 6 million cones and 12 million rods in the human eye. Distribution of the rods and cones varies in different areas of the retina. Fovea has only cones and no rods. While proceeding from fovea towards the periphery of the retina, the rods increase and the cones decrease in number. At the periphery of the retina, only rods are present and cones are absent. First, we'll see the structure of the rod cell. Rod cells are cylindrical structures with a length of about 40 to 60 mu and a diameter of about 2 mu. Each rod is composed of 4 structures: outer segment, inner segment, cell body, and synaptic terminal. This is a figure of rod cells. There's one. Okay. So you can see the cell body, synaptic terminal. This is a nucleus. This is a rod fiber. This is the inner segment. This is the mitochondria. This is the outer segment, and upper one is the disc. Outer segment. Outer segment of rod cell is long and slender, so it gives the rod-like appearance. It is in close contact with the pigmented epithelial cells. Outer segment of rod cells is formed by the modified cilia, and it contains a pile of freely floating flat membranous discs. These are about 1000 discs in each rod. Discs in each rod cell are closed structures and contain the photosensitive pigment, which is called the rhodopsin. Rhodopsin is synthesized in inner segment and inserted into newly formed membranous disc at the inner portion of the outer segment. Now disc push the outer disc towards outer tip. Older discs are engulfed by phagocytosis from tip of the outer segment by cells of pigment epithelial layer. This is how outer segment of rod cell is constantly renewed by the formation of new disc. Rate of formation of new disc is 3 to 4 per hour. Inner segment. Inner segment is connected to outer segment by means of modified cilium. Inner segment contains many types of organelles with a large number of mitochondria. Cell body. A slender fiber called rod fiber arises from the inner segment of the rod cell and passes to outer nuclear layer through the external limiting membrane. In outer nuclear layer, the enlarged portion of this fiber forms the cell body or rod granule that contains the nucleus. Synaptic terminal. A thick fiber arising from the cell body passes to outer plexiform layer and ends in a small and enlarged synaptic terminal or body. Synaptic terminal of the rod synapses with dendrites of bipolar cell and horizontal cells. Synaptic vesicles present in the synaptic terminal contains neurotransmitter, which is a glutamate. Now we'll see the structure of cone cell. Cone cell is a visual receptor with length of 35 to 40 mu and a diameter of about 5 mu. Generally, the cone cell is flask shaped. Shape and length of the cone vary in different parts of the retina. Cones in the fovea are long, narrow, and almost similar to rods. Near the periphery of retina, cones are short and broad, like rods. Cones are also formed by the four parts: outer segment, inner segment, cell body, and a synaptic terminal. So in this figure, we can see the structure of cone cell. This is similar to rod cell. Outer segment. Outer segment is small and conical. It does not contain separate membranous disc as in rods. In cone, the infolding of cell membrane from saccules, which are the counterparts of the rod disc. Photopigment of cone is synthesized in the inner segment and incorporated into the folding of the surface membrane, forming saccule. Renewal of the outer segment of the cone is slow process and it differs from that in rods. It occurs at many sites of the outer segments of cone. Inner segment. In cones also, the inner segment is connected to outer segment by a modified cilium as in the case of rods. Though various types of organelles are present in this segment, the number of mitochondria is more. Cell body. Cone fiber arising from the inner segment is thick and it enters the inner nuclear layer through external limiting membrane. In the inner nuclear layer, cone fibers forms the cell body or cone granule that possesses nucleus. Synaptic terminal. Fibers from cell body of cone leaves the inner nuclear layer and enters outer flexiform layer. Here it ends in the form of an enlarged synaptic terminal or body. Synaptic vesicle present in the synaptic terminal of the cone cell also possesses the neurotransmitter, glutamate. Functions of rods and cones. First we see the functions of rods. Rods are very sensitive to light and have a low threshold. So the rods are responsible for dim light vision or night vision or scotopic vision. But rods do not take part in resolving the details and boundaries of objects. or the color of the objects. Vision by rod is black, white, or in the combination of black and white, namely gray. Therefore, the colored objects appear faded or greenish in twilight. Functions of cones. Cones have high threshold for light stimulus, so the cones are sensitive only to bright light. Therefore, cone cells are called the receptors of bright light vision or daylight vision or photopic vision. Cones are also responsible for acuity of vision and the color vision. It is also called acuity. We'll talk about this later on. What is acuity of vision? Achromatic interval. When an object is placed in front of the person in a dark room, he cannot see any object. When there is a slight illumination, the person can see the objects but without color. It is because at this level only rods are stimulated. When the illumination is increased, the threshold for cones is reached. Now the person can see the objects in finer details and in color. Interval between the threshold for rods and cones, in other words, interval from when an object is first seen and the time when the object is seen with color, is called the achromatic interval. Chemical basis of visual process. Photosensitive pigments present in rods and cones are concerned with the chemical basis of visual process. Chemical reactions involved in these pigments lead to the development of the electrical activity in retina and generation of impulses which are transmitted through optic nerve. Photochemical changes in the visual receptor cells are called the Wald visual cycle. Rhodopsin. Rhodopsin or visual purple is the photosensitive pigment of rod cells. It is present in membranous disc located in outer segment of the rod cells. Chemistry of rhodopsin. Rhodopsin is a conjugated protein with a molecular weight of 40,000. It is made up of protein called opsin and a chromophore. Opsin present in rhodopsin is known as scotopsin, and chromophore is a chemical substance that develops color in the cell. Chromophore present in the rod cell is called the retinal. Retinal is the aldehyde of vitamin A or retinol. Retinal is derived from the food sources and it is not synthesized in the body. It is derived from the carotenoid substances like beta carotene present in the carrots. Retinal is present in the form of 11 cis retinal known as retinine or retinine 1. Retinine 1 is present in human eyes. It is different from retinine 2 that is present in the eyes of some animals. Significance of 11 cis form of retinal is that only in the form it combines with the scotopsin to synthesize rhodopsin. Photochemical changes in the rhodopsin. Wald visual cycle. When retina is isolated and examined in dark, the rods appear in red because of the rhodopsin. During exposure to light, rhodopsin is bleached and the color becomes yellow. When rhodopsin absorbs the light that falls on retina, it is split into retinine and the protein called the opsin through various intermediate photochemical reactions. Following changes occur due to absorption of light energy by rhodopsin. First, rhodopsin is decomposed into batho-rhodopsin, that is very unstable. Batho-rhodopsin is converted into lumirhodopsin. Lumirhodopsin decays into meta-rhodopsin 1 and meta-rhodopsin 1 is changed to meta-rhodopsin 2. Meta-rhodopsin 2 is split into scotopsin and all transretinal. All transretinal is converted into all transretinol by the enzyme dehydrogenase in the presence of reduced nicotinamide adenine dinucleotide. Metarhodopsin is usually called activated rhodopsin since it is responsible for the development of receptor potential in rod cells. Resynthesis of rhodopsin. First, all transretinal derived from rhodopsin 2 is converted into 11-cis-retinal by the enzyme retinal isomerase. 11-cis-retinal immediately combines with scotopsin to form the rhodopsin. All transretinol also plays an important role in the resynthesis of rhodopsin. All transretinol is converted into 11-cis-retinol by the activity of enzyme retinol isomerase. It is converted into 11-cis-retinal which combines with scotopsin to form rhodopsin. All transretinol is also reconverted into all transretinal. Rhodopsin can be synthesized directly from all transretinol in the presence of nicotinamide dinucleotide, which is NADH2. However, the synthesis of rhodopsin from 11-cis-retinal is faster than from 11-cis-retinol. And there is a term called phototransduction. Visual or phototransduction is the process by which light energy is converted into receptor protein in visual receptors. Rhodopsin can be synthesized directly from all transretinol in the presence of nicotinamide. Okay, we have seen the definition of phototransduction. Resting membrane potential in other sensory receptor cell is usually between 70 to 90 microwatt. However, in the visual receptors during darkness, negativity is reduced and resting membrane potential is about 40 microwatt. It is because of influx of sodium and ions. Normally in dark, sodium ions are pumped out of inner segments of rod cells to ECF. However, the sodium ions leak back into the rod cells through the membrane of outer segment and reduce the electronegativity inside rod cell. This is how sodium influx maintains a decreased negativity potential up to 40 microwatt. And this potential is constant and it is also called the dark current. Influx of sodium ions into outer segment of rod cell occurs mainly because of the cyclic guanosine monophosphate, which is called cGMP, present in the cytoplasm of the cell. The cGMP always keep the sodium channels opened. Closure of sodium channels occur due to the reduction of cGMP. Concentration of sodium ions inside the rod cell is regulated by the sodium potential pump. When light falls on retina, rhodopsin is excited, leading to development of receptor potential in the rod cell. Now there is a term called dark adaptation. We'll see the definition of that. Dark adaptation is the process by which the person is able to see the objects in dim light. If a person enters a dim lighted room or a dark room from a bright lighted area, he is blind for some time. He cannot see any object. After some time, his eyes get adapted and he starts seeing the objects slowly. Maximum duration of dark adaptation is about 20 minutes. Causes of dark adaptation. Dark adaptation is due to the following changes in the eyeball. First is the increased sensitivity of rods as a result of resynthesis of rhodopsin. Time required to dark adaptation is partly determined by the time of resynthesis of rhodopsin. In bright light, most of the pigment molecules are bleached or broken down. But in dim light, it requires some time for regeneration of certain amount of rhodopsin, which is necessary for the optimal rod function. Dark adaptation occurs in cones also. Dark adaptation. We'll see the second cause of the dark adaptation. Dilatation of pupil. Dilatation of pupil during dark adaptation allows more and more light to enter the eye. Radiologists, aircraft pilots, and others who need maximal visual sensitivity in dim light wear red glass before entering dim lighted area, because red light of spectrum stimulates the rod slightly while the cones are allowed to function well. This is how the person wearing red goggles can see well in bright lighted area and also can see the objects clearly as soon as he enters the dim lighted area. Now we'll see the light adaptation. The definition of light adaptation. Light adaptation is the process in which eyes get adapted to increased illumination. When a person enters a bright lighted area from a dim lighted area, he feels discomfort due to the dazzling effect of the bright light. After some time, when the eyes become adapted to light, he sees the objects around him without any discomfort. It is the mere disappearance of dark adaptation. Maximum period of light adaptation is about 5 minutes. Causes of the light adaptation. There are two causes for light adaptation. First is a reduced sensitivity of rods. During light adaptation, the sensitivity of rods decreases, and it is due to the breakdown of the rhodopsin. And second is the constriction of the pupil. Constriction of pupil reduces the quantity of light rays entering the eyes. There is a term called night blindness. Night blindness is defined as the loss of vision when light in the environment becomes dim. It is otherwise called nyctalopia or defective dim light vision. Causes of night blindness. Night blindness is due to the deficiency of vitamin A, which is essential for the function of rods. Deficiency of vitamin A occurs because of the following causes. First, a diet containing less amount of vitamin A and decreased absorption of vitamin A from intestine. Vitamin A deficiency causes the defective corn function. Prolonged deficiency leads to anatomical changes in rods and cones, and finally the degeneration of other retinal layers occurs. So retinal functions can be restored only if treatment is given with vitamin A before the visual receptors start degenerating. Electrical basis of visual process. Electroretinogram, ERG. ERG, electroretinogram, is a record of electrical activity in retina. When light rays stimulate the retina, a characteristic sequence of potential changes occurs, which can be recorded in the form of ERG. This diagnostic procedure is useful in determining retinal disorders such as cone dystrophy and retinitis pigmentosa. Acuity of vision. Acuity of vision is the ability of eye to determine the precise shape and details of the object. It is also called the visual acuity. Acuity of vision is also defined as the ability to recognize the separateness of two objects placed together. Cones of retina are responsible for acuity of vision. Visual acuity is highly exhibited in fovea centralis, which contains only cones. It is greatly reduced during the refractory errors. Test for visual acuity. Acuity of vision is tested for distant vision as well as the near vision. If there is any difficulty in seeing the distant object or the near object, the defect is known as the error of refraction. Distant vision. Snellen chart is used to test the acuity of vision for distant vision in the diagnosis of the refractive errors of the eye. And for near vision, there is the chart called Jaeger chart, which is used to test the visual acuity for the near vision. So this was all about the visual process. It is important before we are going for the visual pathway, how we acquire the vision. So this was all about it. Let me know if you are having any query regarding this.